today's video, I will introduce you to how we compute synthetic lethals and metabolic networks based on the concepts of FBA, but using a very efficient algorithm developed in our lab known as the FastSL algorithm. So, we came up with a new algorithm called FastSL in our lab, and today we will discuss this algorithm. The core of this algorithm is that we try to find a different FBA solution to start with and make some intelligent optimizations that helps us cut through the search space. So, instead of solving 170 million LPs, we end up solving a few 100,000 LPs. That gives you phenomenal savings in time and computation power. Okay. So, our approach involves heavily pruning the search space. So, how do we prune the search space? We will get to that. So, we then exhaustively iterate through the remaining combinations. So, it is in some sense related to the exhaustive enumeration, but involves only computing a very small fraction of the exhaustive set. So, we successively compute 3 sets. First we call JSL, the set of single lethals, right. Then JDL, which is if J is the set of all reactions, right. So, J cross J is the set of all pairs of reactions, and JDL is a subset of that. JTL is a subset of JQ, right. Any 3 reactions from the total network. Right. So, the central idea is we use FBA to compute a particular flux distribution, and we use this vector to further simplify the computations. So, we will get to what this vector is in a moment. So, this is our good old linear program, right. What is FBA? Maximize V biomass such that SV equals 0 subject to some lower bounds for all reactions and upper bounds, right. We then identify a flux distribution which obeys the constraints of FBA and also sustains maximum growth, and let us say J that you see on the right hand side is the set of all reactions. The set of all reactions that carry a non-zero flux in a given FBA solution, let us call that JNZ for non-zero. Right. This is a smaller subset of J. In fact, this diagram that you see is drawn to scale for E. coli, the E. coli model that we used. Right. So, how does this help? We have some very interesting observations now, although it seems almost obvious in hindsight. If a reaction J carries 0 flux in the minimal norm solution, in the FBA solution, let us not even worry about the minimal norm solution yet. If it carries 0 flux in the FBA solution which is constrained to support growth, it cannot be lethal. This is a very simple sort of an obvious kind of thing, right. If a reaction is already carrying no flux, removing it is not going to kill the organism. That is very obviously reasonable, right. So, which means there is no single lethal reaction outside of J and Z. That is the next step, next jump, right. So, this is a set of all reactions. All these reactions that are not in this dark portion, they are not carrying a flux. In a given, there are many possible solutions, remember. You remember that, right? There are many possible solutions to any FBA problem, but if it does not carry a flux in any solution, it means that it cannot be a lethal reaction because the cell is growing without that reaction. Now, therefore, we can say that all the single lethal reactions belong in J and Z because these guys are not carrying any flux. So, all the single lethal reactions belong in J and Z, which means that first of all you do not need to do J simulations like size J simulations, you just need to do size J and Z simulations to find synthetic single lethals in the first place, right. So, now okay, so this means that the set of all single lethal reactions is contained entirely in J and Z, fair enough. The next step, let us consider pairs of reactions. So, you can represent them in a matrix form, right, a J cross J matrix, right. Let us consider all pairs of reactions. We can now represent it in a matrix form, a J cross J matrix, and now we say that if a pair of reactions carry 0 flux in the minimal norm, again they cannot be a synthetic lethal pair. That is a very obvious extension of the previous result, right. If they are both, so there are no reactions that are lethal in this orange part. So, both reactions cannot be in J and Z. So, all synthetic lethal, so and this is SL. So, these things will anyway not carry a flux, right. You remove a reaction and along with which is singly lethal, and you remove another reaction from the cell, it is again not going to recover growth in under this formulation. So, all these are anyway known lethals. So, let us leave that out too. So, all synthetic lethal pairs therefore now lie in this narrow region, right, which is again drawn to scale for E. coli. But if you carefully look at it, this is still not a major saving. There are more savings to be done by reapplying the idea and so on, and the gains are even more substantial for higher order lethals. So, let us just look at our results. So, we find that for single lethals, instead of solving about 2050 LPs, we solve only 393, right. So, that is already a 5-fold saving, not a big deal. But for double lethals, instead of solving 1.6 million LPs, we solve only about 8000 LPs, which is a 200-fold saving. And if you look at quadruple, the savings are astronomical, right. So, something that would take weeks would be done in hours. That is the level of simplification we have attained by making this search space slicing that we are doing. So, the smaller the set of non-zero reactions J and Z, the lesser the number of LPs to be solved. Right. So, I did not tell you how we came up with this solution in the first place, right. So, all I said was we have an FBA solution. So, let us just write it out, max V bio such that SV equal to 0 and some LBI less than equal to Vi less than equal to LBJ less than equal to Vj less than equal to Ubj for all j in the reaction set. So, the number of reactions is what we used to call R, right, the size of this reaction set. Right. Now, let us say you got a particular solution V. Now, what is the number of non-zero reactions in V? This is something we need to find out. So, if you recall, can you recall parsimonious FBA? So, in parsimonious FBA, what did we do? We maximized V bio such that we would minimize the L1 or L0 norm of the vector, right. Normally, we want to ideally minimize the L0 norm, that would be the real sparsest vector, but because that also translates to an MILP problem, we prefer to do the L1 norm, right. Once again, what is L1 norm? Right. This is the L1 norm, right, the absolute value of every element in the vector. So, now for the faster cell formulation, does it matter which one we pick? You can even take the regular FBA solution that you get without doing any of these, but the smaller the size of J and Z, faster is faster cell, right, because you get much more savings, right, because what is this, you are trying to make this set smaller. So, let us say you had, if this was J and Z versus this as J and Z, this is going to give you much higher speeds, the smaller J and Z. So, how do you compute the smallest J and Z? You have to minimize the L0 norm or at least L1 norm, which is exactly what we do. We stuck to the L1 norm for ease of computation and speed, but you can potentially use the L0 norm as well. So, the minimized L0 norm solution of the FBA LP problem finds the sparsest solution. However, that requires solving an MILP. We instead use the L1 norm, which translates to this. So, minimize the sum of all fluxes in your final biomass flux such that your FBA conditions are all satisfied, your mass balance, your bounds and so on, and additionally V bio is the same as what you obtained in the first FBA, where you try to maximize. Now, amongst the alternate candidate solutions of the FBA problem, which all give the same function value V by max, can you find the one which gives you which has the smallest number of or the smallest sum of fluxes, ideally smallest number of fluxes, but that being a costly problem, we solve for smallest sum of fluxes. It is possible. So, that is a good point. So, in practice L1 norm works well for us. I would say that ideally we need to go for L0 norm, but because we need, see for we need to do this multiple times during the course of a double lethal analysis. So, you do not want to end up solving so many L0 based problems. So, unless it is a pathological model where the savings are very different between L0 and L1, I do not think this is an issue. So, for all the organisms that we tried, L0 and L1 were not a big deal. There was obviously a difference, maybe 6, 7, 8, 10, but single or low double digits, really no big major difference. So, FastSL finally achieved a 4 times speed up over MCS enumerator, much higher speed up over the earlier reported SL finder, but MCS enumerator is the more recent algorithm, and we also tested this with exhaustive enumeration. And it turned out that the results obviously agreed exactly. We have proof for the algorithm as well. So, a very similar approach can be used to find synthetic lethal gene sets as well, right. The only thing with gene sets is it is a little more trickier, right. Remember that you could have R1 is G1 or G2, R2 is G2 and G3. So, when you remove G1, nothing gets affected, but when you remove G3, R2 gets affected, and maybe even R3 gets affected, right. So, you have to worry about what reactions go out when you remove a pair of genes, and we did discuss this some time back. If you want, you can refresh your memory, but this involves incorporating all the gene protein reaction rules. So, this was our final result table. So, for E. coli we got about 4x speed up on average for double, triple and quadruple gene deletions. So, it took us only about 8.5 minutes to identify triple gene deletions. If you recall the SL finder, it was taking about a week, but of course NCS enumerator takes only about 17-18 minutes. So, the speed up is almost 4x, but the thing is this scales very well. So, we are not sure how MCS enumerator scales, right. It depends upon how scalable the MILP algorithm itself is, whereas in our case we do not worry about, we just have many simple problems to be solved. We have about a few 100,000 LPs to be solved. So, this was the approximate serial time that we took, right. So, it took about like 19 days for tuberculosis, whereas we needed only about like 12 minutes. This took about like 900 days and we needed about 26 minutes and so on, right. But of course, this is serial, so it is not an interesting comparison, but just to let you know that we have done that and basically double verified our claims. So, this was parallel. So, this was across 896 processes. So, this finally took a day for us. So, I mean if you were to put so many processes for FastSL, it will like terminate in like half a second or something like that, because we are still solving a lot of parallel LPs. So, in fact the way the algo works is that we assemble a list of LPs to solve and then like distribute it and solve. The first assembling involves doing some preliminary LPs, JNZ computation and things like that. So, synthetic lethals, I hope I have convinced you that they are difficult to identify computationally because of the combinatorial explosion of possibilities. So, the previous approaches have used FBA to exhaustively search the entire space or pose it as a bilevel MILP. We avoid both. We avoid both the MILP formulation and exhaustive enumeration, but our approach is inspired by exhaustive enumeration, wherein we cut down the search space and finally have about a 4,000 fold improvement for synthetic lethal triplets. We also systematically evaluate gene deletion. The previous studies had only looked at reaction deletions. This involves some intelligent handling of the GPR constraints and things like that, and our results agree exactly with exhaustive enumeration, and we find the applications are in finding functional associations and combinatorial drug targets. You might be able to find some really obscure functional associations, right. So, 3 genes in different pathways could actually be involved together because they involve the production of the same metabolite or something like that in some way. The other thing to remember is that synthetic lethals are environment specific. So, all these were done in a minimal glucose medium, but you can potentially compute synthetic lethals in any different environment. So, in today's video, I hope you got a good overview of the FastSL algorithm, which is a very different take on synthetic lethals in metabolic networks. It tries to slice through the search space by exploiting the alternate optima of FBA essentially, and we get a very good speed up. Almost we end up solving only 1 in 4,000 simulations when you are looking at triple gene deletions and so on. So, it is a very efficient method which can also be easily parallelized to compute synthetic lethals. In the next video, we will start winding up with constraint based approaches. We will first look at the limitations, essentially what are all the things that can go wrong when you predict lethality or growth using constraint based approaches.